I guess I can say Happy New Year and Happy February to everybody, and welcome to this new meeting in the ReMicS network. I think Hanne started saying thank you, but I also would like to say thank you to Hanne for keeping up this network at this busy and isolated time. So I think it's challenging to keep on pushing the microbiota research now, but I think it's really important that we meet and that we exchange ideas and so on. So really thank you for that, and also thank you to Tuula who took the long trip from Finland today to meet us on Zoom. So Tuula has been leading the core facility for proteomics, and we have had some nice collaborative projects already. We are running a regional research network for microbiota research, and Tuula will today talk about a national initiative for advanced proteomic research network. So please, Tuula.
Yeah, okay, thanks. I will share my screen. Now you can see my two cats, and now you can hopefully see my slides.
Yep.
Yeah. So I will introduce you a little on this national network of advanced proteomic infrastructure that was funded by Forskningsrådet. Basically, we made the application in 2018. The funding decisions came in 2019, then we had a negotiation round. So in practice, we got started in the middle of 2020 with the funding. So this is still pretty new. And I thought that probably not all of you are really experts in proteomics, that I'm showing a few introductory slides to what basically proteomics is and how we carry out the experiments, so that you have an idea on how the workflow goes, and maybe get an idea what you can then later on do with your samples. And then I will introduce with few slides our network and some of the new instruments that we are already having. And please just ask questions anytime if you have any. So if we think on proteomics, of course, the word proteome is directly coming from the word genome. And the biggest thing with proteomics is, compared to genome, is that it's extremely dynamic and it's much more complex. So basically, the wording in this slide, the nice wording is that genes only come to life when translated into proteins. Is the big thing. Of course, genome studies are important and transcriptomics is important, but proteomics is still the only thing which is telling you what is actually really happening inside your genes and tissues. And then the wording proteomics is basically the large-scale study of proteins. And here, what is important to understand is that we mostly do non-targeted driven approaches. So we are doing so-called shotgun analysis. We try to identify everything that we have in the samples. Nowadays, also targeted methods are possible, but they are not as common and easy to apply to most of the research questions as the non-targeted workflows. But I think within clinical proteomics context, I think the targeted methods will become more and more important all the time. And if we think on the type of information that we can get from proteomic level experiments, is that we can, of course, identify how much proteins we have in the different samples. And then if we do some type of fractionation, we can go into subcellular distributions, so gain spatial information. Importantly, we can analyze protein modifications and interactions also. And just as a kind of reminder on the complexity of the proteome, it's highly dynamic and it's much more complex than genome. So it's been estimated that after alternative splicing and translation and post-translational modifications, we have more than 1 million different type of proteins in our bodies. So, and we really need to get snapshots of the proteome in a given time, in a given place to understand the full complexity of the proteome. And again, going into the clinical setting, which is probably most relevant to you guys, this is taken from an NIH workshop on clinical proteomics some years ago. So basically, deeper understanding of the human proteome can help fill in the gaps between genomes and phenotypes, and even going into the way of transforming the way we develop diagnostics and therapeutics, and enhance overall biomedical research and future healthcare. And this is something, since precision medicine and personalized medicine is kind of a big thing at the moment, but in this, one big next step could be personalized proteomics. Since, of course, thinking on every individual, we have different proteomes, so we need to understand that on personal level. And I think with the new techniques that we have, new instruments, new sample prep workflows, new data analysis tools, we are closer to getting into this than ever before. And if we have a look on the proteomic kind of workflow and what is required, so we are highly dependent on liquid chromatography, mass spectrometry instruments. So basically these two, which are operated by expert users. And of course, especially this mass spec part is quite expensive. So in most places, like in Oslo, these are centralized in the core facilities. And when you deliver sample to most of the core facilities like ours, we can start out from very many different type of biological samples. Then we extract the proteins. We always digest them into peptides. And then we continue with peptide level measurements with LC-MS and then with the mass spec and data analysis. And how we basically identify the protein by mass spectrometry is that we first measure what are the peptides that are eluting from the LC. So we measure peptide masses, and then inside the mass analyzer, inside the mass spec instrument, we choose, for example, this peptide to be fragmented inside the mass analyzer. And then we measure the peptide fragment masses. And then based on those, if we would get this type of mass spectra, we could basically calculate then the peptide sequence. But in practice, when we do this, we are not doing any type of manual interpretation of the spectra, but we do a database search with the acquired MS and MS data to identify the proteins. And with mass spec, we can of course do other things than protein identification. And most often what we do is we quantify proteins. We have different workflows for that. Mostly we do something which is called label-free protein quantification. So we do relative quantification of the proteins between the samples. And then we can go and identify and characterize different post-translational modifications and characterize protein complexes. And important here is that we mostly do so-called bottom-up workflows. So we start out with peptide level data. So we are not really analyzing a lot on intact proteins when we do mass spec experiments. And then the mass specs and sample prep and data analysis tools have developed immensely over the past 20-25 years ago. Here is an example on a Nature paper from 25 years ago on identifying a protein from SDH page gel, just a standard protein, by using a mass spec workflow. And then you got a Nature paper out of identifying few standard proteins from the gels using the mass spec. And the additional paper from 25 years ago was the biggest proteome catalog until that time, which was 150 gel spots from 2D electrophoresis gel, where they identified around 1,100 proteins in total. Whereas what we can do nowadays is we can do half an hour proteomes. So in this example, there was evaluation of one of the newest instruments in the market in 2018. So identification of around 55,000 unique peptides covering close to 6,000 proteins in half an hour of mass spec analysis time. And now we are, from this instrument, we already have newer generation instruments. So I would claim that with the latest developments in mass spec-based proteomics, we are close to making it possible to analyze single cell proteomics with mass spec. And in Norway, basically, proteomics is centered in 6 core facilities in the different universities. And they offer quite a wide range of proteomic services and expertise for protein analysis. In addition, there is many researchers with focus on proteomics on the side of the method development point of view. So basically, expertise in mass spectrometry, analytical chemistry, biopharmacy. But prior to 2019, we were mostly having quite old mass spec instruments in the different core facilities. And since proteomics is technically very challenging, it was evident that there is more need for collaboration and sharing of expertise in these different units. And this is a picture of a typical mass spec in a proteomics lab. So this is an instrument from Thermo Fisher, and over the past 15 years, this type of Orbitrap high-resolution mass spec instruments have been dominating in proteomics. So I think in all of the proteomics core facilities in Norway, we have had different generations of Orbitrap instruments only, mostly working. And in 2018, we made an application to Forskningsrådet on National Network of Advanced Proteomic Infrastructure. We actually made the first application already with almost the same groups involved in 2016, and then we were graded 7 out of 7, but did not get the funding. So 2 years later, we got the funding, and the official launch was in 2020. So we have, as partners, we have the proteomic core facilities, 2 different core facilities in Oslo, one in Ås in NMBU, one in Bergen, one in Trondheim, and one in Tromsø. And in Oslo, we have also quite many groups with focus on proteomic technology development. In here, you can see our partners. So basically, it's the core facility that I am heading, one core facility in Blinden campus headed by Bernd Tiede, and then we have also Fridtjof Lund Johansen, who is doing mostly antibody-based proteomics here in Oslo University Hospital. Then we have a group led by Stephen Wilson in Department of Chemistry, and a group led by Leon Ropsat and Trine Grønhaug-Halvorsen in Department of Pharmacy. They do a lot of method development for LCMS based assays. And then in Oslo, we have one additional partner, Jens Banke from Department of Pathology. He's not really doing a lot of proteomics as such, but he's really interested in setting up imaging mass spectrometry. And then that is something I will show you one slide on later on. And the funding that we got from Forskningsrådet will be mostly used to purchase cutting-edge new mass spec instruments. And this will, of course, expand the proteomic capabilities of proteomic research in Norway. It will increase basically the analysis capacity, but it will also make novel type of proteomic assays and measurements possible. So at the moment, we have purchased to our network 4 different type of instruments. 2 different instruments from Thermo, so 2 latest generations of Orbitraps, and to Oslo we have purchased 2 different machines from Bruker. And then these are really new in our facility. So 2 weeks ago we started the installation of our timsTOF Flex. So basically the machines are coming from Bruker from Bremen, Germany. So they ordered a delivery truck, and then there was some problems on the way from Bremen to Oslo, since when the truck driver was having a break in Sweden, somebody broke into the delivery truck, but nothing was really stolen. 1 or 2 boxes were slightly damaged, but luckily only from outside. So we got the boxes to Oslo. I managed to get the 2 service engineers from Bruker who are coming from Sweden to cross the border to get to Norway, and then convinced the hospital security that they were allowed to enter our lab. So then we actually got the boxes opened and into our lab. And then we also managed to fit in actually the machine into our lab. So this is a very big instrument compared to the Orbitrap type of machines. So in here you can see that there is approximately 1.5 centimeters of extra space before the roof for the machine. And we think always that mass specs are kind of sensitive and personal instruments, so you need to give a name to the mass spec for it to work well. So we named ours Hilton II. It seems like a royal name to me, and in Finland we don't have any royal family, so I thought that Hilton II is a good name. And here you can see Hilton I. He's a small dog, a friend of mine, and he can smell actually migraine attack. So when my friend is about to have a migraine attack, Hilton can sense it and tell it to her before the attack is actually very bad, and so that she can take her medication very early. So we hope that our Hilton II will be as sensitive as the nose of this little wonder dog. And this is an example what we are able to hopefully to do in a few weeks after we finalize the installation and training of the new instrument. So in the added benefit compared to the instruments that what we are having at the moment is the high speed and sensitivity. The instrument is having an additional dimension in the mass spec separation. So it's not only separating the ions based on mass to charge, like most of the mass specs, but it's also taking into account on their shape. So it separates also according to ion mobility. So we have an extra dimensional separation, which is coupled to very fast type of mass spec analysis, time of flight mass analysis. So we can really do fast measurements with that one. And then this is showing the sensitivity of the instrument. So when we have 1 nanogram of HeLa digest, so just human HeLa cells digested into peptides, from 1 nanogram, we the machine can identify around 1,800 proteins. And when we go to 50 nanograms, we should be able to identify around 5,000 proteins with this platform. And one additional feature which we have not had any possibility before is that this instrument timsTOF Flex is having a dual ion source. So we are able to do basic LC-MS type of analysis, but in addition, it is having a multi-ion source. So basically what we can do with that is multi-imaging. So we can take this type of small tissue sections onto the multi-target and then do the mass spec analysis throughout the basically the slide. So then we can get images like this, which is a lipid image of rat brain. So we can see the differential distribution of lipids and peptides. And this is still limited to quite small molecules, but this is something that we aim to invest more in the future. In the application to Forskningsrådet that we did in 2018, we had dedicated quite a big work package for doing mass spec imaging and dedicated quite a lot of funding. But when we got the funding decision from Forskningsrådet, it was evident that we only got partial funding, not the full funding that we applied for. So we could not afford to buy a dedicated multi-imaging instrument with that funding. But I really wanted to go towards this direction, which I believe will be important in the clinic at some point. So we then, this is a brand new type of instrument, not been available before on the market with the dual ion source. So I wanted to go for that. We needed to push a little on the funds also from the hospital side to make it possible. But now we have it, so we have the possibility to start examining this in the near future on what we can really do with this type of new technique. And thinking on standard nano-LC MS approaches, the nano-LC, which is the thing before the mass spec, which is the one to separate the peptides, is a very important and integral part of the proteomic analysis, but it's also often the bottleneck, since we use so-called nano columns, which are very narrow, 50 to 75 micrometer inner diameter separation columns, and the LC needs to operate in very high pressure, and that causes different types of problems with leaking, wear and tear of the parts, and also problems with carryover and run-to-run reproducibility. So thinking on future clinical proteomics, we have purchased a novel type of LC setup called EvoSep1, which is really aimed for clinical samples, so it can provide very fast, up to 300 samples a day runs, and then it's very robust and still sensitive. So when we can combine this with the new mass spec that we are having, we should be having very good tools for doing large-scale clinical proteomic studies in our facility. Overall, how we see the importance of our network is that we hope to significantly advance the expertise and technology for advanced proteomic research in Norway. And what is really important is sharing of the technology and expertise. So we hope not to be only like an instrument park with new mass specs, but we really spend time and effort on sharing technology and expertise. And then here, the admin manager that whom we have hired for the network is really important. We have already set up a small additional network inside NAPI for advanced data analysis with a member from from each of the nodes in here so that we can discuss and share expertise on proteomic data analysis. And then we have already done one user survey to monitor how happy or unhappy our users in the different places are and how we could develop our kind of services and tools to meet the needs of the users. We have a steering committee, so basically members of the host institutions that whom we meet regularly, and then importantly we have a 3-member scientific advisory board to advise us on how to best develop the network. And our advisory board is Professor Janne Lehtiö from Karolinska. He's also scientific director of SciLife Lab. He's specialized in cancer proteomics. Then we have Ole Jensen, who is professor of protein mass spec from Odense, Denmark, and director of the Willum Center for Bioanalytical Sciences. And then we have Professor Katrin Markus, she's specialized in functional proteomics and director of this proteomic center in Ruhr Universitat Bochum. So they have already given us in quite a lot of advice how they see that we should aim in developing NAPI in the future, and hopefully they can help us also in future funding applications and convince host institutions to invest in the network. And then what is important in NAPI is that we want to have kind of complementary areas of expertise for the different nodes. We cannot all specialize in the same thing. So our basically expertise area is in clinical applications, antibody-based assays coming from Fridtjof, and then later on hopefully also in imaging mass spec. And then the other unit in Oslo is highly specializing in post-translational modifications. We are also quite good in doing phosphoproteomics, but we are not really focused much on other modifications at the moment. So we really aim in increasing this type of node-specific expertise in the coming years. We have pretty nice web pages where you can get more information on all of this. This is just to illustrate on analysis of post-translational modifications by mass spec. So of course PTMs are vital in signaling pathways and in regulating cell function, and then this is one of the most important tasks in proteomics. And in many of the NAPI core facilities we have experience in analysis of well-studied PTMs like phosphorylation, and then these most often involve different enrichment strategies to help to detect low abundance peptides like titanium dioxide or immobilized metal ion affinity chromatography to enrich for phosphopeptides. And then we are quite actively working towards analysis of other PTMs like acetylation, glycosylation, methylation, ubiquitin and ubiquitin-like modifications, and lipidation also. And modomix, so basically epigenetic epitranscriptomic modifications is something where we have expertise in only one of the core facilities. So researchers in NTNU, in PROMEC, they have extensive experience in analyzing these modifications by MS. And then through NAPI funding, they will soon install new LC chromatography instrument that will facilitate highly accurate RNA modification profiling. So this is our webpage where you can get a lot of information on what we do. We are also active in Twitter. So, and for any type of contact and inquiries, you can contact either Joe, who is our admin manager. He's also PhD in proteomics, so he's quite experienced on proteomics in general. And then you can, of course, contact me. Or when you want to start out a new project, you can go directly to your local core facility, and in case they feel that somebody else is better qualified for your project, they can help you contact the correct node. And like you've probably got an email, we are organizing a small kickoff seminar in March, Friday the 5th, from 9 to 12. We were a long time hoping that we could do some small physical symposia either in Oslo or somewhere else in Norway, but now it seems that it's still a long time ahead before we can do any physical meeting. So we decided that we do the kickoff meeting now when most of the units have already purchased new instruments, and we can introduce them a little bit more in detail and the possibilities that they give us. And then one additional slide which might be relevant to this audience is that now UEO Life Science is having an open call for third time for convergence environment, and then their emphasis is on antimicrobial resistance, sustainable lifespan, neuroscience, personalized medicine, and enabling health technologies, which are kind of directly related to the new life science building. And I placed there an abstract with title Proteomics Goes Personal, basically proposing an interdisciplinary project utilizing the new instruments to exploit the power of proteomics to address challenges in personalized medicine. And I think this is something we could maybe discuss a little if there is any common interest towards this type of application also within this network. It doesn't have to happen now, but there is still 1 month or so before we need to make the first draft for this type of application. And then I think this was my last slide. So if you have any questions, I'm happy to answer, or we can discuss in general the network.
Okay, thank you very much, Tuula, for this nice overview. I thought I could leave the questions section. If you have questions, you could either put them in the chat or try to raise your voice. I was thinking, just as a starting point here, many of us may have studies or projects or phenotypes that where proteomics becomes relevant. And what you have not said so much about is perhaps kind of the types of material that you like to investigate. And typically, for instance, what is very readily available are materials like serum or plasma, which I've understood often represents the big challenge for proteomics. Could you say something about that?
Yeah, I think we prefer everything but serum and plasma. But we can analyze serum and plasma, but they are a challenge for proteomic analysis because they are so abundant with albumin and few other proteins which are always dominating in the analysis. And then there is the possibility to remove with different type of depletion columns. Very easy to use spin columns, for example, to deplete, for example, 14 most abundant proteins. We have tested them quite extensively coming from different vendors, especially the spin columns, which are very easy to use and would be suitable for kind of large sample sets. But they seem to have problems in not doing the depletion reproducibly. So we have very different amounts of albumin, for example, left in the samples after the depletion, and then when we do the LC-MS, if we have very different amounts of albumin left, then doing the kind of relative quantification between the samples becomes a challenge. As we first normalize before we do the analysis, we inject the same amount of peptides into LC-MS, but if there is like tenfold difference in how much albumin we are actually injecting, then the quantification becomes a problem. So I think this is something we still need to work a lot on. And I hope with the new mass spec, we could also, because I think there is also, in addition with serum samples, there is the ongoing discussion in clinical proteomics field that if we use any type of depletion, what other than albumin, for example, we are depleting at the same time, since albumin is very sticky. So very many low abundant proteins stick to the albumin and then they are non-specifically being depleted. So in that way, the preferred option would be analyzed directly intact plasma or serum. And then basically we end up identifying around 200, 250 proteins from plasma only. So I think this is something that is like the least preferred starting material. Everything else is more simple for our approaches. This was actually a very important question, since there are thinking on plasma analysis, which is of course very kind of important and interesting in the clinical setting. There are other workflows than mass spec based assays which can then be used for plasma sample analysis, but then they are always kind of more like targeted approaches. But there are things like SomaScan, which some of you probably have heard on, but it's only commercially available that you need to buy it as a service. But then there are some other platforms that if we would get extra funding, for example, for the network, we could afford to buy and start using for this type of samples.
Okay, thank you. I have one more. Marius, you had a
Yes, it's just a question in line with the one with Johannes. So if you were to plan a clinical trial today and you wanted to focus on bacterial proteins, what would be the since plasma and serum are difficult, what would be the best samples to obtain in your biobank to get good and reliable proteomic results? Would it be cells or
cells are easy. Tissue samples are easy. Then saliva is something that.
Sorry. Go on.
Yeah, saliva is something that we can work quite nicely with, but I don't know if it's with, maybe actually with bacterial that could even work. But saliva collection, of course, is not, I guess, straightforward in many ways. So I think there is a lot of things that we should kind of be working together on designing the experiments starting from the very beginning, so that they are kind of feasible in practice for sample collection, which is important part in any of these, that the samples are collected and stored in the best possible way, and you know that they are not lying around in the benches in room temperature for longish time, and what is feasible.
Right. One.
I can just follow.
Gregor also has a question,
yeah.
I think.
Okay, good. Yeah, sorry.
All right, thanks, Marius. Are any of your machines capable of running native mass spec?
The ones that we purchased, they are not optimized for native mass spec, and I think the one in Tromsø, I would need to check on the details what they actually ended up in their full package, what they included, but native mass spec is not really among the kind of the prioritized areas, since, like I mentioned, we only got partial funding for the network, so we could not afford all of the things that we had planned to purchase. But we already, last fall, we did the second round of application to Forskningsrådet with hope that we would get more funding to kind of complement the instruments that we have purchased.
Okay. Do you think that the native mass spec is something that will be particularly applicable to clinical samples?
I think at the moment, at least, it's so challenging technically that I don't see it as the first techniques. I think probably something like middle down is more applicable, so that we would do partial digestions and analyze like larger peptides.
Okay, thank you. Brian, I think you have a question.
Yeah, thanks, Johannes. Tuula, congratulations on the funding and establishing your team. I think it's always nice when you get these new instruments and you very nicely illustrate how they are important in increasing the sensitivity. My two questions are, one, is there a workflow set up for sending samples to these other centers? So say we're researchers in Oslo, but we want our samples analyzed by a group in Trondheim. Are we meant to send our samples individually, or is there something established already in the network that we can use? And then my second question is more about using the MS facility not only for proteomics, but for instance, metabolomics or lipidomics. Will any of the centers be set up so their mass spec facility or machine can look at these other interesting molecules? Thank you.
Yeah, okay, I will start with the first one. So we hope to set up kind of an at least semi-automated platform where you can send your samples through our web pages or some other tool, but at the moment it's not available yet. You are free to contact any of the nodes if you have, for example, if you got interested in the ReMicS platform in Trondheim, you can just directly contact them, and in case you don't really know whom to contact, you have like a general proteomics question and you don't know who would be best to answer, then I would advise to contact our admin manager, Joe, who knows all of the nodes and can then kind of sort out the best option for your question. I think that at the moment is the easiest option. And with metabolomics, lipidomics, other whatever omics that could be done with mass specs, we are proteomics network. This is the basically with the machines that we are having, we could do metabolomics, for example, no problem with machine-wise, but with expertise and knowledge on sample prep and how to set up the instruments for this type of analysis and everything, we don't have that. And at the moment, I think there is not really like a well-functioning service unit for metabolomics in Oslo. There is a lot of need for that, and that's clear for the mass spec people in Oslo, both on the university and on the hospital side, but how to kind of best establish that there are people doing metabolomics measurements, yes, with mass spec, but not like a metabolomics core facility at the moment.
Yeah, thank you very much, Tuula.
Okay,
but I think also the metabolomics people did send an infrastructure application last fall to Forskningsrådet. So of course, if that gets funded, that will change the situation a little. But I think even if that gets funded or not, I think there is a clear need in Oslo for metabolomics facility.
Thank you. Murat, you have a question?
Thank you, Tuula. And first of all, congratulations for getting this new machine. So really great MS with new possibilities. Therefore, I have a lot of quite specific questions. Perhaps you can in the future organize some new meeting with detailed description of possibilities of deep stuff. What exactly this machine can. But my question, in your opinion, because here is microbiota people, so people interested in microbiota. What do you mean? Is this timsTOF MS, is it suited for metaproteomics analysis?
Yes.
What do you mean?
Yeah. Basically, the group in OS in NMBU, they are doing mostly metaproteomics, and they also purchased timsTOF Pro. They didn't. So basically, it's otherwise the same machine than what we are having, but it's not having the multi-source. They also, we did a lot of testing with their samples when we were evaluating the instruments, and they also felt that this is definitely the best option for their applications.
Very good. To follow up on that, can you, okay, one thing is to do mass spec on colony of one bug, but what about the mix of bacteria or even like a stool sample? Would it be relevant to investigate that in the mass spec, or would that be a mess?
That's basically this is a mess not for the mass spec itself, because the mass spec doesn't know where the peptides are coming from, and the mass spec doesn't care where the peptides are coming from, and whether they are known peptides or not known peptides or whatever, but it's a problem for the data analysis. In metaproteomics, I think one of the biggest challenges is on the data analysis. So we are basically dependent on the databases, so proteins which are in the databases. And with metaproteomics, of course, if we would include in the search all the bacteria that are available in whatever databases, in addition to human proteins and everything, then the search base is very large. And then the search is extremely slow, and then there is high risk of false positives. So I think this is something that we should then be working on how to optimize the data analysis. It's not really on the kind of course, depending on the starting material, the sample prep needs to be optimized, but the mass spec part is exactly the same. So it's challenges on the data analysis side. And I think this is one of the benefits of the network, that we can more closely collaborate, for example, with the group in NMBU doing metaproteomics in different research questions already, who are having expertise on this type of data analysis.
Very good, thank you. Gregor, you had one more question?
Yeah, thanks. I just want to say that the NAPI organization you've got sounds very similar to what we have on the sequencing front. So I'm working for one of the sequencing nodes based here in Oslo, but the NorSeq consortium is both geographically and organizationally very similar to what you've now set up for proteomics. So I was very interested when you said that there's a possibility for convergence here. So I think something that could cover both proteomics and also the genetics of any topic would be a fantastic candidate there. What would be essential here is to narrow things down. So since we have an audience here of microbiome researchers, if anyone is interested in, if that rings any bells, you think that could be a nice opportunity, we'd be very happy to discuss that.
Yep.
Very good. I guess you have listed for this speed dating event.
Yeah.
The convergence environment.
No, I haven't, because we're scrambling to do COVID sequencing at the moment. So dating's off the menu at the moment.
That's too bad. Okay, but I think it's on February 18th. It's the first, and there's one in March as well.
Yep.
Okay, very good. I think, are there other questions or that we should take before the meeting's coming to an end? Do you have an announcement, Hanne?
No, not really. I will keep distributing news for the network, so please just send me whatever it is you want out there. I will distribute the dates for Tuula's kickoff, the 5th of March. We are working on the new meetings for this spring, so we have a lot in store. So
That's very good. Well, I think, thank you, Tuula. I think you gave a very good overview of your network, and there are lots of opportunities. So, and
Thanks.
it's obvious that we just contact you then probably,
Yeah.
and with proposals and for discussion and quick questions.
Yeah. Good.
Very good. I
thanks
think
again,
that's.
Tuula.
So then, thanks again, everyone, for joining the meeting, and hopefully we'll see each other again for a meeting in March.
Yeah.
Okay.
Bye.
Bye, everyone.
Okay, thank you, everyone. Bye.